Hello everyone. Today we are talking about the importance of the cell membrane and how it lets certain things in and out of the cell in a very specific way. This is a close-up of the cell membrane found in your textbook. You see the layer, this wrapper, this cell membrane that wraps up the cell, is actually a double layer of lipids. That's why it's called a lipid bilayer. In fact, each lipid is attached to a phosphorus ion, and therefore it's a phospholipid bilayer. Phosphorus, lipid, similar to fat, and bilayer, two layers. So you have the outer and the inner layer. Through the membrane, there are some larger holes. These are protein channels or channel proteins. All right, a protein channel is a channel through a protein, and it is made from a channel protein, a protein that makes channels. Okay, same thing. On the surface of the cell membrane, there are some carbohydrate chains, and these carbohydrate chains pick up messages from the rest of the cell. For example, a hormone might come along, and this carbohydrate chain would detect the message from the hormone. The cell membrane is even more complicated than this, but this is a basic picture of what's going on. Even though the cell membrane is much thinner than, say, a cell wall in a plant cell or a capsule from bacteria, the cell membrane is so important to all cells. In fact, plant, animal, and bacteria cells all have a cell membrane. The cell membrane determines what gets in and out of the cell. It's kind of like a gatekeeper. So there are two types of transport when things get in and out of the cell: passive transport and active transport. If you're passive, you're just kind of not resisting. So passive transport does not require energy. It happens all on its own. However, active transport, the cell has to actively bring things in or out of the cell, and that uses energy. So there are three basic types of passive transport. The first one is diffusion. Diffusion is the process by which molecules spread out from areas of high concentration to areas of low concentration. For example, perhaps this is a beaker of water and we put a drop of food coloring in it. At first, the food coloring is very concentrated here and not concentrated here. In fact, there is no food coloring here. The food coloring will spread out until it's equally distributed throughout the water, until the food coloring reaches equilibrium with the water. The food coloring or any chemical will always move from areas of high concentration to areas of low concentration until all the concentration is equal. That difference is called the concentration gradient. High concentration here, low here. Therefore, there's a gradient. It gradually changes. Well, here I guess it gradually changes. There it kind of changed very quickly. But as it spreads out, then you have very concentrated, medium concentrated, and not very concentrated, a gradual change. And finally, when you're done, you no longer have a concentration gradient. It's all equal. You have equilibrium again. I'd like to illustrate the process of diffusion. Here's a beaker with some water, and I'm going to add a drop of food coloring. You'll notice that the food coloring slowly spreads out inside the beaker until all of the water in the beaker is equally concentrated with food coloring. So the food coloring is moving from areas of high concentration, where you can see the blue, to areas of low concentration, the clear areas. And we'll speed up this process, and you'll be able to see what happens after it has sat still for a while. Just like going down a slide, going from high to low doesn't take any energy. You just let go and down the slide you go. So if you're moving from high to low concentration, you just go down the hill And that happens all on its own. However, if you climb back up the slide, that takes energy. It can be done, but you have to put in energy to get it to happen. That would be like active transport. And in this case, taking the molecules and shoving them back up in a corner, that wouldn't happen on its own. That would definitely require energy. So that would be an example of active transport. So back to the passive transport. A difference between concentrations in a space is a concentration gradient. And when molecules move down the concentration gradient from high concentration to low concentration, then that is a passive process and does not require energy. The thing that is diffusing or spreading out may or may not pass through a membrane. It's called diffusion if it spreads out whether there's a membrane or not. However, a selectively permeable membrane, like the cell membrane, may be in the way. Or, for example, like an air filter. An air filter that you put in your return air vent is a semi-permeable membrane. The air can get through, but hopefully the dust is trapped. So it is permeable to air. Air can permeate it or get through it, but the dust hopefully does not permeate that semi-permeable membrane, your air filter. So the cell wall is a much finer filter. It filters out much tinier things than dust. And it is selectively permeable or semi-permeable. They both mean the same thing. Some things can get through, some things can't get through. So the cell is selective about what it lets through and only allows some things to pass through. Water and oxygen are two examples of tiny molecules that can easily move across the cell's membrane by diffusion. Osmosis is the diffusion of water across a membrane. Let me give you a little more details. First of all, osmosis is a type of diffusion. The particles spread out from high concentration to low concentration. But then it must be water molecules. Only water molecules can undergo osmosis. If it's a different molecule, it's just plain diffusion. Then the water molecules must spread out and cross a semi-permeable membrane. Then you finally have osmosis once all three of those criteria are met. So water diffuses across a membrane. Now, let's say you have water and salt water. Which one is the water more concentrated? Now, yes, there's more salt over here, but we're not asking about that. Which one is the water more concentrated, more pure? This one has more water because it's just water. This one has salt and water, so the water is watered down by the salt? Salted down by the salt? Who knows? So we have more concentrated water in the pure water and water that is diluted by salt over here in the salt water. So if you put the salt water together, then the water would spread out into the salt to try to push the salt away and water down the salt. Okay? It's as if the salt is sucking the water over to it. Now, the salt does not suck up the water, okay? That's not what happens. But it's a good way to remember it. Think salt sucks, the salt sucks the water over there, and the water moves from where there's a high concentration of water to a low concentration of water. Even though there's a high concentration of salt, we're looking at the water. So this is why when you drink a salty liquid, you get thirsty. When salt gets into a cell, the water in your blood also tries to go into the cell to water down the salt. Therefore, you get thirsty because you have less water in your bloodstream. Now let's look at some different types of solutions. We are going to look at isotonic, hypertonic, and hypotonic solutions. First of all, isotonic. If you have two solutions, say with salt, and they both have equal amounts of salt, they're equally salty, equally concentrated, then those are isotonic solutions. The prefix iso means the same. For example, you've probably heard of an isosceles triangle, where the two sides are the same length and the same angle. So an isosceles triangle, iso has the same sides, equal sides. So an isotonic solution has equal concentrations. If you put a membrane between those two solutions, the water will simply transfer back and forth at equal rates, but the overall big picture will stay the same because the concentrations are the same. So here is a picture of an isotonic solution. We have a 10% solution inside this semi-permeable membrane and a 10% solution of salt outside. Therefore, the water just moves in and out of the cell in equal amounts. When one water molecule moves out, the other water molecule moves in. Next is a hypotonic solution. Hypo means low. Hear that? Hypo, low. For example, if somebody is hypoglycemic, they have low blood sugar. And a hypotonic solution has low amounts of dissolved salt, sugar, food coloring, whatever you are dissolving. It's got lower amounts of it. So for example, this solution is hypotonic, low, compared to the water inside the cell. So we have 20% salt inside the cell and 10% salt outside the cell. Therefore, the solution outside is hypotonic. The solution inside the cell is hypertonic. Hyper means high. Like if you're hyperactive, you have high amounts of energy, right? So this is a hypotonic solution with high amounts of dissolved stuff, in this case, salt. So remember, salt sucks. There's more salt here, less salt here. So the salt sucks in the water. So the water moves into the cell. Now, is the salt really sucking in the water? No. That's just a fast way of figuring it out. Here's what really happens. If there's 10% salt, there's 90% water. And if there's 20% salt, there's 80% water. Now, water is going to move from where there's more water to where there's less water. Oh, so the water moves into the cell to try to water down the extra salt. So water moves from high to low concentration into the cell. The salt is too big to fit through this semi-permeable membrane. If it could fit through, the salt would move out of the cell. But since it can't go through this semi-permeable membrane, water can permeate, salt cannot, and therefore the water has to move to go inside the cell until finally these are about the same percentage of salt. Next, let's look at a hypertonic solution. As we've already explained, hyper is high, like hyperactive. So in this case, once again, the solution outside the cell has high amounts of salt and low amounts of salt inside the cell. So let's take a look at what happens. All right. We have 10% salt inside the cell and 20% salt outside. So the solution is hypertonic, hyper amounts of salt, and the cell is hypotonic, lower amounts of salt. But it's the water that moves. We have 80% water outside and we have 90% water inside. Therefore, you move from high amounts of water to low amounts of water, so the water moves out of the cell. Once again, the salt cannot pass through that semi-permeable membrane, so it remains where it is. Now, back in this hypotonic solution, the water moves into the cell, and the cell will get bigger and bigger and bigger. In fact, in an animal cell, there is only a cell membrane, which is actually fairly delicate, even though it's super important. An animal cell placed in a hypotonic solution may eventually explode because of the amount of water that enters the cell. A plant cell, however, has not only a cell membrane, but also a cell wall. So the cell wall is sturdy and strong enough to prevent a plant cell from bursting. So you can put a plant into 100% water and have no problems with the plant cell bursting because it has that cell wall to provide the extra stability. But an animal cell does not have a cell wall, and it could easily burst. So something like a paramecium, remember these things, the parameciums with the little cilia, little hairs all around them? A paramecium will often have a contractile vacuole. It's a hole that it pumps full of water. And then finally, when it's full of water, it squeezes it. So instead of looking like that, it squeezes it down to a little spot, and you can see the little wrinkles around it that look kind of like a star or a flower. And it squeezes this hole shut and squirts the water back out. So that way, a paramecium can live in fresh pond water. And even if the water keeps flowing into the paramecium just pumps it right back out, and it's perfectly fine. Your cells don't have that, but you have skin around your body to prevent you from getting waterlogged. So what if a cell is placed in a hypertonic solution? Well, the water will travel out of the cell and the cell will shrivel up. So if you put a plant in salt water, the plant will wilt and fall over. And also, if you put an animal cell in salt water, it will eventually shrivel up and die. In fact, this is how salt kills slugs. The salt dissolves in the slug slime on the outside of the slug, making a hypertonic solution. So the insides of the slug are like the cell. There's less salt. The outsides of the slug are like the solution with more salt. So the water moves out of the slug, and the slug dehydrates and dies of dehydration, and that's how salting a slug kills it. Crazy, huh? Right. So diffusion and osmosis are both types of passive transport. Passive, sitting there letting it happen, no energy required. So there is a third type of passive transport that uses those carrier proteins. Remember those from the picture at the beginning of these notes. Those carrier proteins act like a little tunnel, a protein channel, and that allows big molecules to go through. So here's the phospholipid bilayer, and here is a protein channel. So it's kind of like a tunnel that lets things through this cell membrane. See, water and oxygen are so tiny, they can actually fit between the phospholipid bilayer of the membrane. So oxygen and water are so teeny-tiny, they can just zip right between and get right through the cell membrane, which is a good thing because you want oxygen and water to be able to get to the cell really easily because your body needs it. But bigger molecules like sucrose, sugar, need a way to get into the cell because they're too big to just squish between the cell membrane parts. So instead, sucrose or other molecules will come in through a protein channel, or a channel protein, or a carrier protein. It's all the same thing. And it will just drift through that large hole instead of squishing through the cell membrane itself. That is called facilitated diffusion. Facilitated means helped. Okay? For example, the MC for the Oscars facilitates the Oscars program. They don't do the whole Oscars program. They introduce the people announcing the name, they introduce the performers, they facilitate the Oscars program and make sure it runs smoothly. So a facilitated diffusion, it's diffusion, it's spreading out from high to low concentration, but it's facilitated. It's helped to go more smoothly because it goes through this protein channel. It's still passive transport because no energy is used. Next, there are several types of active transport. Active means it has to actively push or pull things in or out of the cell, and that requires energy. The reason is because those substances are going against a concentration gradient. Instead of going the high to low concentration, this is like going up the slide, you have to go from low to high concentration. So for example, with the salt in the cell, there is already more water inside of the cell than outside. So if the water packed in and made even more water inside the cell, that would be weird. That would not be normal. And that would require active transport. You'd have to have something that could push the water in, because it's not going to want to go there naturally. Things that do active transport include a protein pump. This is a protein pump. Part 1, Part 2, and Part 3 of what's happening. It looks a lot like a protein channel. It looks kind of like a little tunnel. But there's a little gate inside. See how it kind of squishes together? And when a molecule that the cell wants comes up to the protein pump, the protein pump pushes the molecule through. See how it changed there, the shape changed? It pushed the molecule through and into the cell. Because there would have already been several molecules inside the cell and fewer outside, and the molecule wouldn't naturally move into the cell. So a protein pump is what does this molecular transport. It transports the molecules through the pump into the cell. But what if the molecule is so big it can't even make it through the pump? That's when the cell will do either endocytosis or exocytosis. Endo, enter, exo, exit. Cyto, cell. So endocytosis enters the cell. And for example, an amoeba uses endocytosis to eat little paramecium or whatever. So the amoeba stretches out its pseudopods, its false feet, and surrounds the particle of food, the paramecium, the euglena, whatever it is that it's eating, and wraps it around and reattaches the cell membrane around the outside and makes this into a vacuole, a storage hole. Then it brings more lysosomes with digestive particles and empties them into this vacuole, and that digests the food. So that is endocytosis when it enters the cell. There are two main types of endocytosis, phagocytosis and pinocytosis. Phago means food, so the cell is eating food, just like you saw with that amoeba. Then, if the cell brings in a liquid, pinocytosis is the cell drinking. And so the cell can take in liquid that way as well if it doesn't want to wait for the water to diffuse through, or if it needs more water than it would naturally have. The opposite of endocytosis, of course, is exocytosis, where something is exiting the cell. And this is how a cell gets rid of waste products. Remember those vesicles created by the Golgi body? Those aren't waste products, those are proteins that it needs to send to another cell. And so it uses exocytosis. The little vesicles go up to the surface of the cell, attach and link to the surface, and then spill out of the cell. Those vesicles can go into the bloodstream. It also takes the lysosomes that have digested the cell parts and then perhaps have waste products, and it will spit those out of the cell as well. So the sodium-potassium pump in your body is a very famous example of a protein pump or molecular transport. You know that it's not good for you to have too much sodium in your diet, but you do need some sodium, just not a lot. Then your body also needs more potassium. So it wants to get rid of the sodium and take in potassium, and it actually does this all as one process, like this. This is the inside of the cell and this is the outside. Right now, we have a bunch of sodium ions. Na, capital N, lowercase a, is the formula for the sodium ions. So these sodium ions go into this sodium-potassium pump, this protein pump, and they stick inside in the little holes. Then the pump flips and spits those sodiums out of the cell into the bloodstream. Then your cell needs more potassiums. So 2 potassium ions, K is the abbreviation for potassium. So these 2 potassium ions go into the pump, and the pump flips again and pushes the potassium into the cell, which is pretty cool. So for every 3 sodiums pushed out of the cell, 2 potassiums are pumped into the cell. And that's how the sodium-potassium pump works. Notice on the last page there is a list of words that you should know. Be aware that you will need to know which ones are active transport and which ones are passive. For example, diffusion, osmosis, and facilitated diffusion are all passive transport. They do not require energy. Then, endo and exocytosis, as well as protein pumps like the sodium-potassium pump, are all active transport. Now, a carrier protein is used in facilitated diffusion, and phagocytosis and pinocytosis are both types of endocytosis, so those are all active transport. Finally, ATP is an energy molecule. It's kind of like a rechargeable energy battery made from a molecule instead of a battery. What happens is ATP stands for adenosine triphosphate. Tri, 3, phosphate, or phosphate ions. So, what happens is the adenosine triphosphate molecule, when it's charged up, like a battery is charged up, it has all 3 phosphorus ions. When the battery, the adenosine triphosphate battery, gets used up, it gives away one of those phosphate ions and becomes adenosine diphosphate. Di means 2. Then it's like a dead battery. So then you recharge the battery by adding that phosphate back to it. Now you have adenosine triphosphate again, and it's like a charged battery, and it can be reused to provide energy to the cell. So when we say ATP, we mean energy, ATP energy. And it's energy stored in the ATP molecule.